All right, it's already past 4. If Dr. Lefkowitz, if you're ready, I think we can get started.
Okay.
Um, let me. All right, we started recording. That's great. All right, good afternoon, everybody, and welcome to our first bioinformatics event in this semester. And thank you all for joining us today. We will be discussing bioinformatics and health informatics resources and tools available here on campus. And I hope that the seminar will be quite interactive and that maybe you came with your own questions related to your own research, and hopefully you will get them answered. And our speaker for today is Dr. Elliot Lefkowitz, who is a professor of microbiology and also director of bioinformatics in the Center for Clinical and Translational Sciences. Dr. Lefkowitz has been a director for bioinformatics core facilities since 1992, and he has a great experience in assisting researchers at UAB with their own data analysis that requires some bioinformatic skills. And speaking of Dr. Lefkowitz on research interests, he's interested in microbial evolution and microbial genomics, as well as the role of microbiome in human health and disease. That's great. And with that, thank you. First of all, thank you, Dr. Lefkowitz, for coming today and for giving this overview talk for us. And with that, you can take it over and we can get started.
Thank you. I'm happy to be here today. Let me start off by saying I want to make this very informal, so please interrupt me if you have any questions, and I certainly hope to have lots of time at the end for questions as well. If you do have a question, please just unmute yourself and speak up, because I'm not going to be able to see you with the limited number of panels that it's giving me right now. But my goal today is to talk about bioinformatics resources on campus. And let me start off giving you my contact information. So if you have any questions subsequent to this talk today, please feel free to contact me. The only thing that's out right now is you'll never find me in my office, except maybe late on Sundays watering my plants. But otherwise, please feel free to contact me any other way. So I'm going to talk about the services that are provided by the UAB Center for Clinical and Translational Science, the CCTS, specifically in terms of its informatics unit. Now, I'm also, as you'll see, I'm going to expand out into what else is available at UAB, but I'm director of the Bioinformatics Service Corps for the CCTS. And we provide bioinformatics services to UAB investigators and also to other CCTS investigators who are members of some of the other institutions that are part of the CCTS. The CCTS has sort of a hub and spoke model in which UAB acts as the hub for all CCTS activities. But then there are a number of other institutions in the Southeast, from Hudson Alpha to Southern Research, Auburn, LSU, Tulane, Southern Alabama, a number of other institutions are part of the CCTS. And the CCTS has a general goal of providing research resources that can enhance and promote translational research. And informatics is one part of that. CCTS informatics encompasses both clinical informatics types of resources that they provide, as well as bioinformatics. Clinical informatics helps give investigators access to clinical data from the electronic health record, helps promote clinical research in various ways. If you look on the homepage now, you'll see access to clinical data on COVID-19 cohorts. That is a national effort. I'm only going to talk today about the bioinformatics resources, but be sure you should be sure to take away that if you are interested in working with patient data or some other form of clinical research, that those resources are also available, and you can either go to the CCTS page under Research Common, and there'll be contact information there, or certainly feel free to contact me, and I can put you in touch with the necessary people. But as I indicated, I'm going to talk about bioinformatics. Now, CCTS is not the only unit on campus that can provide bioinformatics support. We work with a combination of a number of different units. One is the Heflin Center for Genetics or Genomics, or the Genomic Service Corps, that has sequencing capacity and can do a lot of the sequencing, next generation sequencing that people might need on campus. And then also David Crossman provides bioinformatics support for various projects. And so he is also a source of support. The Cancer Center, through its biostatistics shared facility, also provides bioinformatics support. And we work closely with their bioinformatician, as we do with David Crossman from Heflin, but we also work with Dongquan Chen, who's the bioinformatician that's part of the Cancer Center. And so each of those has core facility like resources that are available to investigators at UAB. Beyond that, sort of ancillary support that is closely related to bioinformatics support certainly includes biostatistics. And here, biostatisticians, both from the Department of Biostatistics in the School of Public Health, as well as the Department of Medicine, the Division of, what is it, Preventative Health Medicine? I'm going to get that wrong. But anyway, they're biostatisticians that also work through the CCTS, the Biostatistics, Epidemiology, and Research Design, called the BIRD unit. And so they can also provide their expertise to lend a hand in study design, and frequently will take bioinformatics analysis up to a certain point, and then you might require more advanced biostatistics in order to look at the significance of different data sets and differences in those data sets. And BIRD can help out with that. We work closely with UAB IT Research Computing. They provide Chiaha, which is a source of subsidized, you don't have to pay for most Chiaha resources that give you the high performance computing facilities that are needed to do a lot of bioinformatics analysis. And so we work with them to ensure that those resources are available to support bioinformatics, and then also working with them to provide some of the education and training that's needed to utilize those resources. After that, a number of other departments have bioinformaticians who can provide bioinformatics services to people in those departments, but also many times people outside of those departments. In the Civitan Center, Laura Ionoff is available. Biochemistry has Yvonne Edwards. And then there are other bioinformatics faculty certainly spread throughout campus. The Informatics Institute is one primary place in which bioinformatics faculty from across campus, but also some of the bioinformatics faculty who have appointments in the Institute, such as Jake Chen and others. Zhe Chen can provide support as well. And then, of course, faculty in other departments. I'm sure you may be familiar with Liz Worthy or Brittany Lassen. All of them are available to some extent to provide support, but at least they're doing research that involves utilization of bioinformatics and also involves development of new bioinformatic tools. And so that base of knowledge, that expertise, is something that we want to share across all investigators at UAB. So something we've been talking about in the last year or so is really to develop a new bioinformatics core, something called the Distributed Bioinformatics Analytics Core, to provide analytic support for bioinformatics. But the key here is that word distributed. And that word distributed means we want to take advantage of all of these other resources that are available on campus and make it easier for individual investigators to take advantage of their expertise. We want to just make all of that easier. Okay, but I'm going to specifically talk about CCTS bioinformatics today. And I'm going to start off talking about some of the general resources that we provide, some of the general opportunities that are available to support your analytical needs. And then I'm going to show you examples of just a few of the projects that we've supported over the last few years. But first and foremost are the people that are available to do that support. So currently, our bioinformaticians comprise 4 individuals: Dongquan Chen, who's out of the Cancer Center, and then directly part of CCTS are Curtis Hendrickson, Travis Potacek, and Liam Vanderpaul. All of them are full-time bioinformaticians who are available to support people's needs. A number of people I should also mention in the past who've developed or helped to develop a lot of the resources that we have available. They've been there for important components of what you see today, even though they aren't currently here. Samantha Carlisle, Ranjit Kumar, Jordana Mwanguzai, and John Osborne have all been part of our effort in the past. And then beyond just bioinformatics support, of course, we need a lot of computational support. And so we share with the Informatics Institute and informatics architect Jailai Wang, who developed some of the key resources that I'll mention in a bit that support sort of the basic data management needs of a core like ours for bioinformatics. And then a programmer, Don Dempsey, does a lot of the basic programming support that we have. Okay, so overall, the types of services, the types of resources that we provide are direct analytical assistance. The first 8 hours are subsidized through the CCTS, and then that assistance does need to be compensated either by putting a bioinformatician on a grant or by a fee-for-service basis. We just track the amount of time and effort expended, and then the grants charged based upon the time that was spent on a particular project. These frequently involve more detailed research collaborations. That is, I think we're most successful when we become part of the laboratory's research team. So not only getting involved once you've generated the data, but actually getting involved before you even start to do the experiments. That is, when you're thinking about the overall study design, because we can help a lot there based upon our knowledge of the sequencing state of the art, so the types of sequencing resources that can be utilized, or biostatistical resources. We can bring a lot to the table in terms of helping in the original study design. But certainly then we stick around and we're there as the data is being generated. We can make suggestions and help out a bit with data management, and then certainly getting access to the data for performing the analyses that are needed. Access to software is something, analytical tools. So we'll install tools on Chiaha, and so those tools, once installed on Chiaha, are available to you. There is this platform called Galaxy, which is a web-based interface to doing biostatistic or bioinformatics analysis. Galaxy is a nice platform. There's a nice public Galaxy that is available. I think it's called usegalaxy.org that you can go to. We do provide one from here, and we have for a number of years. One thing which we realized early on is it takes a significant amount of support to keep it running and up to date. And right now, all of our bioinformaticians have 100% of their effort devoted to actual bioinformatics projects and not supporting Galaxy. So Galaxy is a little bit out of date. I hope if that distributed bioinformatics core can come to fruition, that will then enable us to provide the resources needed to update Galaxy and make it really usable on campus. Certainly access to data, databases, analytical tools, pipelines. So pipelines that stick together multiple tools that are needed to perform any sorts of analyses are important. And so all of this we're providing, and the mechanism that we're providing that is through UBright. You may have heard of that. That's something that the Informatics Institute is supporting, ubright.org. And that's a place where UAB investigators can go and get access to these resources. a lot of this we're doing in concert with all of the other bioinformaticians on campus and the groups that are also developing their own pipelines. So all of us use, for example, RNA-seq pipelines to evaluate and analyze data from transcriptomic experiments. And it would be dumb to waste resources by everybody developing their own particular analysis pipeline. So we're trying to work together to combine resources and get much more efficient in developing these resources and then certainly making them available to everyone here. I already talked about access to computational resources in Chiaha, and then I'll also talk a little bit about the training and education that's available through our core. This just shows you UBright. This is the front page to UBright. So you may want to come here and just explore the page a little bit. Certainly there's a lot here now, but I expect there'll be a lot here in the future as well, a lot more here in the future. Okay, to talk a little bit about education, not all of you may realize it, but now we have an undergraduate bachelor's program in bioinformatics that started in 2018. I'm co-director of that program with John Johnstone, a faculty member in the computer science department. And so, you know, we've really just had 2 classes so far. We have 9 students, and I think that's going well. And some of those students will be taking their first bioinformatics classes about a year from now. We also have a master's program in that has a concentration in bioinformatics. This is the Master of Science in Multidisciplinary Biomedical Science programs. Their URL is here if you're interested. And so that's a general master's, but it has a number of different concentrations, and bioinformatics is one of them. And then, of course, there's the PhD program that's part of GBS, the Genetic, Genomic, and Bioinformatics theme. And so here's their URL. And certainly a lot of the courses and such that are available come about because of the existence of this particular theme. And then UAB Research Computing provides regular programs to educate people on how to utilize Chiaha. And so their URL is right here now. And in addition, my group, then there's workshops and seminars that occur throughout the year. If you look at the various newsletters that come out, you can see those being announced. And then CCTS Bioinformatics, my group, provides in-person one-on-one training to do whatever it is you'd like to do. So when we collaborate with a lab, when we help a lab analyze their data, one of the first things we ask is, okay, do you have a student or a postdoc who'd like to do some of the analysis themselves? And if they do, we'll try to work with them in order to train them to the extent necessary to help them analyze their data. If those students or fellows want more advanced training, then they could go ahead and take maybe some of the master's programs or classes or the PhD classes as well. So there are lots of different ways to get training and education in bioinformatics. Okay, so the software is all great. The education you need to understand and utilize that, but we also need an underlying infrastructure to support what we do, especially to manage the data that we work with. And so I mentioned Jailai Wang, our informatics architect, who's helped a lot in the last couple of years that he's been here to develop this infrastructure that supports what we do. And so one is, a lot of you may be familiar with version control of code. So you have a version control system. Git is the system that most people use today, GitLab, and that's used to store your code and update your code, check it out, utilize it, make changes, check it back in. Well, we're using GitHub or GitLab to do data version control. So not only code, but data. So all of the data we work with gets packaged up and then gets pushed into a GitLab repository. And then that is further backed up currently into Box, eventually to something else, but that gets backed up as an archive of all of the data. And so that really has allowed us to create a separate GitLab project for every data analysis project we work on for every investigator. And I can't tell you how much of a mess it was in the past before we had this to track down where the data was for any particular project. And this now makes it relatively easy. And the process of doing all this is automated. It's sort of a one word piece of code that you just type in at the command line, and you automatically get things pushed into GitLab and then backed up and such. We have various tools for tracking all of our projects and effort devoted to the projects. I mentioned Don Dempsey, one of our programmers. He's been involved in development of Kaizen. Some of you may have interacted with Kaizen, which is an educational platform to support various different types of education. It's really exploded. James Willig is the one who spearheaded Kaizen and its development here on campus. And then there are various other analytical platforms that are being supported by UAB Research Computing that also are available to support a lot of our data analysis needs. One, if you need a virtual machine, if you need to establish a virtual machine to run some new code and you don't want to screw up someone else's computer to do it, or if you have a project that's long running, multiple people need access, you don't have a server that can support it. Well, now Research Computing provides OpenStack, which allows you to create virtual machines, and those can be provisioned. And again, it's all subsidized through UAB IT and UAB itself. And so that's available to everybody. And then access to the high performance computing resources, Chiaha, is available through Open on Demand, which is a nice web interface to allow you to work with Chiaha. So to get you beyond as necessary, just solely working on the command line. There's certainly a lot of what we do and a lot of what you may do is going to be carried out on the command line, but there are other ways to do it too. And so Open on Demand is something that you may want to check out. Okay. And then, as I mentioned, we have development of pipelines. It's collaboration between a number of groups. A lot of those pipelines utilize SnakeMake, which is a scripting language, and we've developed pipelines along with others to support transcriptomic analysis, genomic assembly and variant detection, microbiome and metagenomic analysis. A lot of this we're now starting to use for SARS-CoV-2 investigations. We utilize Conda-based packages for dependency management. This is a way to package up in sort of a scriptable format all the tools that you need to obtain and install different small programs that allow you to manage the dependencies. Anybody who used to install a program from the command line on a Linux system or a Unix system in the past realizes what it took to ensure that all the dependencies were there. Conda-based packages make that easy. And easiest still is to put everything into software containers, Docker containers, which puts everything, including the operating system, into one small package. And so we're doing that for our microbiome analysis pipeline, for example. But this is also available, and we'll be doing it for other things. And these packages run on Chiaha as well. And this just to show you some of our activities. We try to track our activities in any number of ways. I think the most useful one to show that we actually are making a difference is tracking the number of publications that have come out of our work, as well as the citations to that work. So the publications are publications from the people, from the investigators that we're working with. And if we have certainly close collaborations, then some of the CCTS bioinformatics staff are part of the authorship on those publications. And so in blue, you'll see the growth in publications. In green are the publications in which the CCTS bioinformaticians aren't necessarily authors, but these are coming out of the investigators lab that we support. So you can see it grow from the initiation of CCTS informatics in 2008 to present. We have well over 550, almost 580 publications right now. And then citations to those publications is even more impressive, with well over, what is it? Windows blocking. I think that says over 10,000 right now, citations to all of these publications. So that just gives an example of the amount of what comes out of what we do. Okay, so now I want to switch gears a little bit and really start to talk about just a few examples of what we do. But are there any questions up to now? Just speak up.
I think we have a question in the chat
Okay.
about the person training one-to-one. Is it available to all UAB students?
Yes, anybody can just contact me, certainly. Or if you go to the CCTS webpage I showed you, it's called We're Under Research Commons, and there's contact information there. And so just indicate that you'd like assistance with something. And then what I'll do is put you together with the bioinformatician who can best meet that need. Okay, so what do we do more specifically? So we certainly work with a lot of next generation sequencing data. And that can be whole genome sequencing data, in which we're going to assemble and do variant analysis of that data. We've worked with human model organisms, anything from mice, rats, ferrets, anything you can think of. A lot of bacterial microorganism whole genome assembly, viruses, including SARS-CoV-2, but other viruses as well. And in addition to doing variant discovery, we also have worked looking at disease phenotype associations with sequence variants. Excuse me. A lot of gene expression work, whether it be RNA-seq analysis or looking at various aspects of transcription regulation using ChIP-seq or methyl-seq or ATAC-seq. We've done that. Microbiome analysis is a large component of the services that we offer, and this is through the microbiome core that's run by Casey Morrow. And in addition, we also do metagenomic analysis of microbial populations. Again, that's run through the microbiome core facility. But that's just a few examples. There's a lot more types of different projects that we participated in. And so what I want to just show you is a few of the projects and a few of the publications that have come out of this work. So the microbiome work, as you see here, is supported now by Liam Vanderpool and Dongquan Chen. Travis Potacek, who's here now, also has supported a lot of microbiome work, and a lot of this was started originally by Ranjit Kumar in my group. And so what we're just doing, of course, is looking at the microbial population of humans and then also of model organisms where we can actually experiment with the types of associations that and the types of causative effects microbiome populations may have. What you do is you have your sample, you extract the DNA, it's sequenced using primers specific for the 16S ribosomal RNA gene. Those are sequenced, and then we analyze it in order to generate a readout of all the bacteria that are present on the basis of specific 16S gene signatures, sequence signatures that allows us to identify, usually down to the species level, what organisms that are present. So the first thing we did when UAB just started to get involved in microbiome analysis was develop an analytical pipeline. I think we're up to 244 runs, and each run has multiple projects. So potentially close to 1,000 projects or more that we've worked on with microbiome projects over the years. And so this is a summary of the pipeline that we've developed in order to analyze this sequence data. It runs on Chiaha. It's something called QRAP, Chime Wrapper. Chime is a package developed by another group, Rob Knight's group, that provides a set of tools that are utilized to do microbiome analysis. QRAP uses Chime tools. It also uses some other tools in order to do the analysis. So there's the basic analysis that involves quality control, clustering all of the reads, the sequence reads that are present into clusters of similar sequences. And then those sequences are categorized according to what taxonomic group they belong to. That is, what bacterial species or genus or higher taxonomic levels that they belong to. Then we take that data and do a variety of statistical analyses to determine the significance of the different experimental groups that the investigator was using. And then we provide a visualization of that. And this just shows you the website that we provide, in which we provide access to for each individual investigator who has submitted a data set for analysis. And so here you have the different links, and you get a link to a bar chart of each sample along the horizontal axis here. And then here in the colors are the different organisms that were identified as a result of our analysis. This happens to be a large study. Not all of the studies we do have quite this many samples. But statistical analysis, such as like alpha diversity, so what is the diversity of organisms within a sample, and then beta diversity plots, looking at the diversity of organisms present between different samples. So each dot here represents a different distinct sample, each sample which has its own microbial population. So the closer any of these two points are, the more closely related their microbiome population. Now you see here, now we're just sort of color coding it by individual samples. That doesn't help you at all. You can't really see specific groups. But if we overlay information on top of this plot, now we start to get more important information that may point to whatever it is that the investigator is studying. So here we simply overlay male and female, male being in blue, female seeing here, and you immediately now see separate groupings of these points. And now we could overlay additional information dependent upon where what this study was originally supporting. So that's an example of the microbiome analysis. Here are just a couple of papers. One, we did a paper, and this was in collaboration with Christina Musney, who's in infectious diseases, and also people from LSU, Chris Taylor, and others, to work on optimizing the conditions that we were using for doing microbiome analysis. And there are a variety of different conditions. The primer you use, whether it's the variable 4 region of the 16S ribosomal RNA gene, or variable 1 to 3 or 3 to 4, you can choose different ones. These are looking at vaginal samples in a study of bacterial vaginosis. And here we were interested in optimizing which primer to use and also optimizing the sequencing center. So we're comparing sequencing at UAB here to sequencing at another center that gave us data for all of these other primers. And our readout was the number of unique taxa that were identified and the number of individual species that were uniquely identified. And the bottom line was UAB did the best in terms of getting the sequence. This was done by the Heflin Center. So it did the best in terms of generating data that gave a better data set than the other sequencing center. And also our default V4 region was also the best here. Then just to give you an idea of the specific results from this bacterial vaginosis study, again, this was a study funded and the PI was Christina Musney. Just one figure from that paper looking at one particular case of bacterial vaginosis, just following over time that patient over multiple times. This right here in red, a Nugent score, is an indication of an outbreak. And what you see here with an outbreak is a shift of bacterial species from commensal species that are more lactobacilli species. So these are all lactobacilli species leading up to an outbreak of BV. And then during the outbreak of BV, those disappear, and then you get pathogenic species. Gardnerella is one. Adipobium, Prevotella, those are here. And so this is the type of study that we've been collaborating on with microbiome analysis. Okay, let's go beyond the small microbes into the humans and model organisms themselves. A variety of the bioinformaticians, Curtis Hendrickson, Jordana, Samantha, and John Osborne, have worked on various studies. This is just a simple alignment of various sequence reads to the human genome sequence. And so this panel up here just shows the read depth across this region of human chromosome 14. And so what we're interested in is the variant. So this is this small region of human chromosome 14 here. And you can see a variety of variants in these colored bars in this read depth chart point to variants with respect to the human genome reference. Sometimes those variants, like in this blue line here, are homozygous. All you see is that variance. Many of these variants you see are heterozygous, seeing two alleles represented at that particular position. And we can zoom in even more and see at this particular allele, the reference is a G, but we also have an A present in almost half of the samples. Statistically, it's probably half and half between A and G. And so that's what the variant detection shows us. Of course, what we're really interested in is associating variants with some phenotype or some disease. And so what you do, you have a cohort of patients in which you're sequencing those patients. You have then your control patients, and you sequence that cohort as well. And so you're looking for a particular variants that appear only in your experimental cohort and not your control cohort. And one way of visualizing that is from a gene burden test here, in which these are representative of variant alleles, and you're looking for a peak here as an indication of a predominant variant in that particular region. In this case, it looks like of chromosome 6. So human genetics, now we can go to bacterial genomics. And this stems, this is sort of, in addition to microbiome studies, we're interested in the behavior of microorganisms in any number of different ways, especially as it comes to human disease. Travis Potacek, Liam Vanderpoel, and also, again, Curtis Hendrickson have participated in various bacterial genomic studies. This is one particular study. The PI was Michael Neerweiss in the microbiology department, in which they were interested in looking at iron utilization by Mycobacterium tuberculosis that causes TB. And so what they did is they did a transposon mutagenesis study, randomly knocking out genes in the MTB genome, and then looking for particular knockouts that affected iron utilization. And so this study identified genes and therefore identified strains of these knocked out genomes that responded differently to iron. And visualizations like this can help to understand what associations there might be between specific genes in this organism and particular aspects of iron utilization. A lot of work in transcriptomics. Curtis, Samantha Carlisle, and Travis have worked on this over the years. Here we're doing RNA-seq studies in which we generate reads that in this case map only to the exonic portions of genes here. So that's where you see your read depth only within exons. Here's a study that Samantha worked on with David Standard as the PI, looking at Parkinson's patients, looking at gene expression in monocytes, and in particular, looking at gene expression in females versus males. And as you can see here, here's Parkinson's status here, with controls being green, cases being red. So here's cases, or I'm sorry, here's controls, here cases. You can see a very a different profile in in these monocyte expression genes here between cases and controls. In males, it's less pronounced. And in addition, the genes are all different. So this is an indication that Parkinson's disease is a very different beast in males versus females, at least when looking at gene expression in monocytes. And so this study is ongoing and being followed up. The paper for this study has just been submitted or resubmitted based upon an initial set of comments. And then finally, I want to end up today with something that's certainly relevant to everything going on now, is looking at what we're starting to do with SARS-CoV-2 sequencing. And this involves collaborators Kevin Herod and Sixto Leo. And so initially, as you can imagine, a lot of different labs are doing studies with live virus. And so Kevin Herod is providing live virus to investigators at UAB for their studies. And one of the first things you want to know is the sequence of the virus that you're providing them with similar to or the same as the sequence of the virus that's infecting human beings. Um, and since you have to passage that virus in order to grow up enough to do the experiment, there's always a concern that there might be changes. And indeed, when we do the sequencing, we can see those changes. First of all, this is a visualization of what we sort of commonly call our contamination pipeline. It's based on a tool called Corona. We run this now for any next generation sequencing data set that we work with, and it quickly looks through the reads and it tells us what those reads may map to. So in this case, this is from the SARS-CoV-2 virus sequencing project. About half of the reads map to coronavirus-like or specifically SARS-CoV-2 sequences. So that's good. These are samples coming from human samples, either blood or nasopharyngeal swabs. And so you don't expect everything to be virus. You expect some to be human. And so these are the human or human-like sequences that come out. There are some bacterial sequences there and some things that Corona didn't identify. I couldn't tell you offhand what they are, but how you run the analysis and how you handle the reads, this may be more or less. But then we go and we assemble all of the viral sequence reads to the reference sequence for SARS-CoV-2. And so we get a very nice assembly that crosses, that assembles against the whole surface of the SARS-CoV-2 genome. You see here, though, that there seems to be a peak in reads down towards the 3' end of the SARS-CoV-2 genome. That actually is expected because SARS-CoV-2 not only produces a primary genome that covers the whole genome from beginning to end and a primary transcript that covers a good part of the genome, but it also provides a number of subgenomic transcripts down here. And that's how these messages and proteins end up being transcribed and translated. And so we expect to see more reads down here because you get these subgenomic transcripts. So this is showing us just what we expect. You can see some variants starting to pop up here. Some of these variants you see here are in all of the reads, and therefore they're variants throughout the viral population. Those are just the normal mutations that we expect to see as the virus has been passage from human to human from its start in China throughout the world now. And so you see these small mutations accumulate. But then when we actually look at the mutations in the virus stock that's being produced, we start to see a few low frequency variants. So you see here, these variants run from anywhere to 0.6 to 8.1% of the reads. So they don't yet predominate in that stock of virus, but we do see some significant deletions. Interestingly, these are all still in frame deletions. So this may actually be producing virus that's viable in the tissue culture stocks. Then we also see some SNPs. And so what people are now seeing, investigators around the world in producing these virus stocks for their own labs, you see these mutations accumulating in that polybasic cleavage site of the spike protein that's part of the attachment site of the virus to the cell. And so that does seem to change as you passage it in tissue culture. So that's a concern. So now they're trying to approach growing these virus stocks in a different manner to minimize the accumulation of these particular mutations. Okay, so I'm going to stop there and I'm going to leave it open to questions in general. I'm going to stop my share so I can see you and you can see me. And then let me start here with a couple, let's see. Okay, what's in there is just a thanks. So unmute yourself and ask a question as you may be interested.
I guess I'll start. So does CCTS provide any fellowship grants for students? If so, what about citizens and non-citizens?
So CCTS does have a training program that provides pilot awards. Those are more directed at fellows and faculty members at UAB. So they don't provide student fellowships per se. Um, I'm just trying to think through. Yeah, I think that's true. It's always worth checking, but there are other opportunities for student fellowships as well. Um, Certainly training grants and such. There are training programs. The CCTS does provide training programs, and those are available to students. A lot of that is directed at students who may be more clinically or medically oriented and participating in translational research. So those training grants are available. Whether or not they're available to basic science students, I'd have to check on that and get more information.
Thank you.
Thank you. Yeah, and thank you, first of all, for your presentation. It was wonderful. And I had a quick question about the Chrono software.
Mm-hmm.
So you use it to clean up the data a little bit to remove some maybe not contamination, but
We use it to identify what reads are there. So, for example, if someone is interested in SARS-CoV-2 and we run Corona and all we see are human reads and bacterial reads and there's no viral reads, then we go back and tell them immediately something's wrong.
all right. But can it be used for RNA sequencing data too?
Yes, and we use it for RNA-seq data. We're running that on any experiment generating sequence data. We're now running that contamination pipeline first before we do anything else because it saves a lot of time. Because unfortunately, not every data set that you get back is what you think it's supposed to be. And the sooner you can find out that you need to rerun the experiment, and the sooner we can find out so we don't do an analysis that's wasting everybody's time, all the better.
I can totally relate to that point. Thanks, Dr. Lefkowitz, for this wonderful lecture. So I am a fourth year PhD student in biostatistics. And then one of my problem right now is finding data set to use for my methodology work to kind of verify that my methodology works. So I was just wondering, do you have any recommendation for places I should to start to look into data sets for myself? So my primary interest is microbiome as yours. So I was just wondering, is there any public data source that is good to use, i.e., I don't have to do a lot of data cleaning or kind of like upstream analysis or bioinformatics processing for the data set?
Yes, I mean the Human Microbiome Project has generated a large data set, both of microbiome data, but also of metagenomic data, and sometimes the same samples are run on both. So we've used that ourselves in order to test our pipelines, in order to test new tools that we've developed. So I would look up the Human Microbiome Project data sets that are available. Otherwise, if you have specific needs, certainly related to microbiome, you could either ask me or Casey Morrow. Casey is familiar with all of the data sets that have been generated here at UAB. And so if your particular needs happen to match those of a data set that's been sequenced and analyzed here, Casey can talk to the investigator and see if they might let you utilize their data set. But I would certainly first go and look at the data sets that you can get publicly from the Human Microbiome Project.
Got it. Awesome. Thank you so much.
Any other questions, comments?
I might have one more question. So CCTS kind of develops a lot of tools. So is there any training from the computer science side for developing tools and probably machine learning or something like that?
So you're saying training in how to do development of, for example, let's say snake make pipelines.
Yes.
So I would say that that is something we would very much like to be able to do. Right now, with the limited number of people, it's hard to get into that aspect of it. Certainly, if you're a bioinformatics student, some of the classes that you have or will be taking may get into tool development. I don't know if the one that's taught by Malay Basu and Alex Rosenberg, I think it's Info 501, 601. I don't know if they go into pipeline development or not, but certainly they have you learn R and a little bit of Python. If you want to do it casually, you could certainly take some of the online courses in R or Python. Maybe there's something in SnakeMake, I don't know. Computer science department offers classes. If you get stuck, let's say you're trying to do it and you get stuck at a certain point and want someone with expertise in SnakeMake to help you out, or whatever it is you're working on, then you could contact us and we could try to get you over that little hump. But that's something that I would hope eventually this distributed bioinformatic analytics core could also offer, that is a more robust training program that can get into things like pipeline development, container development, Conda packages, that sort of thing. Because as you all may know, if you're certainly doing bioinformatics as a part of your educational program, eventually fellowship and such, you just don't analyze data. You're also a developer. You're also IT support. You have to do a little bit of all of this. And sometimes that lesson comes hard. Sometimes you like it, and it's fun to do. But sometimes you just want to start working with the data, and you want everything to work 100% right off the bat. It never does. It never does. And you're going to get these error messages when running a program. You're going to have to chase down a dependency. You're going to have to figure out which line of code has a small error in it, which data set has a small formatting problem. That's part and parcel of what you're going to be doing as a bioinformatician.
Thank you so much.
Anything else? All right. Well, thank you. And I guess you're going to distribute the notes that contain all of my slides. So those will be available if you want to refer back, and it has my contact information and all of that.
Yes, sure. We will be distributing the PDF file, and also we have recorded the meeting, so we will be uploading it to our YouTube channel, so you could maybe revisit some points if you're interested. And yes, thank you all for attending. Thank you, Dr. Lefkowitz.
Sure, happy to do it.
Great.
Goodbye.
Goodbye.
Thank you. Bye.
Thanks.